Bismillahirrahmanirrahim. We shall explain you today about the chemistry of the dopamine, and we shall also explain you Parkinson's disease. So dopamine is also called as 5-hydroxytryptamine, or dopamine is also called as dihydrophenylethylamine. Now in this context, today we shall explain you about the introduction of the neurotransmitters and the dopamine. Then we shall explain you about the synthesis of the dopamine, where is dopamine synthesized and how is dopamine synthesized. And after synthesis, where is dopamine stored and how dopamine formed in the cytoplasm is transported in the vesicular granule for storage. And then we shall explain you about the release of the dopamine from the storage granule, and then we shall explain you about the receptors of the dopamine, and we shall explain you about the reuptake of the dopamine, and then we shall explain you about the degradation of the dopamine. And in the end, we shall explain you about the functions of the dopamine, and we shall explain you dopamine imbalance, that is deficiency of the dopamine leads to Parkinson's disease, and excess of dopamine causes schizophrenia. So these are some of the contents of our today's lecture. In this context, as far as the introduction of the neurotransmitters and dopamine is concerned, dopamine is a neurotransmitter like acetylcholine, norepinephrine, epinephrine, serotonin, GABA, glycine, all these are neurotransmitters. So along with this, dopamine is also a neurotransmitter. And what are neurotransmitters? Neurotransmitters are actually neurochemicals. And what are neurochemicals? Neurochemicals are biomolecules like carbohydrates, like proteins, nucleic acid. These neurotransmitters are also biomolecules, but these biomolecules, they participate in the neuronal activity or they influence the neurons. Neurochemicals, they include neurotransmitters. They also include the substances which transport the neurotransmitters. Neurochemicals also include neurotrophins. What are neurotrophins? These are the growth factors. Then neurochemicals also includes neuropeptides like endorphins, enkephalins. Now, what are neurotransmitters? As you know, neurotransmitters are group of substances that are released on excitation from axon terminals of the presynaptic neurons of the central nervous system or the peripheral nervous system, and they travel across the synaptic cleft, bind to receptors on the post-synaptic cell membrane where they affect electrical charges, where either they increase sodium permeability, results in depolarization, or they result in the excitation or hyperpolarization. Now, in this context, dopamine is the neurotransmitter of the central nervous system. Dopamine is major transmitter of nerves that interconnects nuclei in the basal ganglia. The term basal ganglia refers to brain's region that includes caudate nucleus, putamen, globus pallidus, substantia nigra, and subthalamic nuclei. These structures, they are located deep within the core of each cerebral hemisphere. Now, what is basal ganglia? Basal ganglia is the part of the brain that regulates movements. Basal ganglia in turn depends on certain amount of dopamine to function at its peak efficiency. The cells which synthesize and utilize dopamine are called as dopaminergic neurons. And dopaminergic neurons are found in number of areas in the brain, chiefly in the ventral tegmental area of midbrain, and they include substantia nigra and are present in the arcuate nucleus, which is arc-like structure in the hypothalamus. And what is substantia nigra? What is meaning of substantia nigra? Substantia means substance of which tissue or organ is composed. And nigra means black substance. These are the layers of melanin containing neurons. Then the process by which dopamine acts as neurotransmitter is divided in the 5 steps. In the first step, we shall explain you about the synthesis of the dopamine. We shall explain you how is dopamine synthesized and where is dopamine synthesized. And after synthesis, how is dopamine transported and stored in the vesicular granule. And in the second step, we shall explain you about the release of the dopamine from the synaptic vesicle in the synaptic cleft. And then we shall explain you about the dopamine receptors, and we shall explain you the binding of dopamine to postsynaptic receptors. And then we shall explain you about the inactivation of the dopamine by enzymes, or we shall explain you the removal of the dopamine. And in the end, we shall explain you about the reuptake of the neurotransmitters. So in this way, the process by which dopamine acts as neurotransmitter, it is divided in the 5 steps. So in the first step, we shall explain you about the synthesis of the dopamine. Now dopamine, it is so named because it contains a single amino group. Dopamine is so named because it contains single amino group, and this amino group is derived from the amino acid tyrosine. So actually, this dopamine neurotransmitter, it is formed from amino acid tyrosine. Amino acid tyrosine is actually precursor of the dopamine. We have already explained you about the classification, about the properties, and about the functions of the amino acid tyrosine. This amino acid tyrosine, this is phenol group containing amino acid. This amino acid tyrosine, it is non-essential amino acid because this tyrosine, it is synthesized in our body. It is synthesized in our body from the amino acid phenylalanine. Phenylalanine is a benzene ring containing aromatic essential amino acid, and this phenylalanine, when it is hydroxylated, when OH group is added in the amino acid phenylalanine, it is converted into tyrosine. And this tyrosine, it is standard amino acid. Like other amino acid, tyrosine is also included among the 20 amino acids which take part in the formation of the protein. So tyrosine is standard amino acid. It has genetic code and transfer RNA, and for that it takes part in the protein synthesis. And this tyrosine, it is also classified as glucogenic and ketogenic amino acid. That is tyrosine. It is called glucogenic because on catabolism it can be converted into glucose, and it is called ketogenic because it can be converted into ketone bodies. So actually, when tyrosine is catabolized on catabolism, tyrosine is converted into amino acid fumarate, and fumarate is the intermediate of the citric acid cycle. And all the intermediates of the citric acid cycle, they are glucogenic. They can be converted into glucose. Similarly, tyrosine on catabolism give rise to formation of acetyl-CoA. And acetyl-CoA is the precursor of ketone bodies. Acetyl-CoA is converted in the liver in ketone bodies, in acetoacetate, in beta-hydroxybutyrate. And these acetoacetate and beta-hydroxybutyrate, they are considered as alternate source of energy in the extrahepatic tissues. Then this tyrosine, non-essential amino acid, it is also required for the formation of thyroid hormone. It is also required for the formation of T3 and T4. Thyroid hormone is found from tyrosine and the iodine. And this tyrosine, it is also required for the formation of melanin pigment, which gives color to the skin. This melanin pigment is found from amino acid tyrosine in the melanocytes. And this tyrosine, it is also precursor of catecholamines. Catecholamines includes dopamine, norepinephrine, and epinephrine. So in this way, this tyrosine, it is the precursor of dopamine. It is precursor of norepinephrine and epinephrine, which are collectively called as catecholamines. So actually, this dopamine, it is intermediate in the synthesis of norepinephrine. And these catecholamines are formed from the tyrosine by 4 enzymatic reactions. However, synthesis of dopamine from tyrosine, it is 2 enzymatic reaction process, while formation of norepinephrine from tyrosine, it is process of 3 enzymatic reactions, and formation of epinephrine, it is process of 4 enzymatic reaction. So in this way, the dopamine is important because dopamine accounts for more than half of the catecholamines production in the brain. Dopamine, where is dopamine found? Dopamine is actually found in the dopaminergic neurons. And where is norepinephrine found? Norepinephrine is found in the adrenergic neurons in the brain. And it is also found in the adrenal medulla as epinephrine. Epinephrine is found in the adrenal medulla. Now, dopaminergic neurons cannot synthesize norepinephrine and epinephrine because the enzyme number 3 and number 4 enzymes, that is dopamine hydroxylase, which is required for the formation of norepinephrine from dopamine, is missing, is absent in the dopaminergic neurons. Or the enzyme which converts norepinephrine into epinephrine, that is methyltransferase, enzyme number 4, is also missing in the dopaminergic neurons. So actually, dopaminergic neurons, they are equipped with the 2 enzymes. They are equipped with the enzyme number 1 and number 2. Dopaminergic neurons are equipped with amino acid tyrosine hydroxylase and dopa decarboxylase, which are responsible for the conversion of tyrosine into dopamine. So in this process, now this dopamine, it is formed in the, where it is found, we told you it is found in the dopaminergic neurons. Where is dopamine formed in the dopaminergic neurons? It is found in the cytoplasm of the dopaminergic neurons. So site for the formation of dopamine from the tyrosine, it is the cytoplasm of the dopaminergic neurons. So in the first step of the dopamine synthesis, this dopamine, it undergo process of hydroxylation. There is addition of hydroxyl group, and this hydroxyl group is added by the enzyme tyrosine hydroxylase. So tyrosine hydroxylase catalyze addition of hydroxyl group in the amino acid tyrosine to convert it into L-dopa. Dopa is dihydroxyphenylalanine. And this enzyme, tyrosine hydroxylase, it is also called as tyrosinase. And this enzyme, tyrosine hydroxylase, it is copper-containing metalloenzyme. This enzyme requires tetrahydrobiopterin as hydrogen donor, and this also requires iron. And this enzyme also requires coenzyme NADPH2, which is required for conversion of dihydrotetrabiopterin into tetrahydrodiopterin. So NADPH is required for reformation of tetrahydrobiopterin by the enzyme reductase. And this tyrosine hydroxylase enzyme, it is rate limiting enzyme, and it is site for feedback inhibition of catecholamines. In the next step, tyrosine, after hydroxylation by the enzyme tyrosine hydroxylase, in the next step, by the action of enzyme dopa decarboxylase, which catalyzes the removal of COO group, carbon dioxide or carboxyl group, on decarboxylation. This dopa means dihydroxyphenylalanine is converted into dopamine. So formation of dopamine from the tyrosine, it is series of two reactions. If we simplify it, tyrosine first undergoes hydroxylation followed by decarboxylation, results in the formation of dopamine. Now, this tyrosine, which is converted first into dopa, now all the dopa which is formed from tyrosine is not converted into dopamine. It is observed that approximately 8% of the dopa synthesized in the sympathetic nerves is not converted into dopamine, but 8% of the dopa, it escapes in the blood. Therefore, plasma level of dopa dihydroxyphenylalanine provides information about the activity of enzyme tyrosine hydroxylase, which we told you is a rate limiting enzyme, which is regulatory enzyme. Changes in the plasma level of L-dopa also occurs in variety of disorders. Patients who suffer from neuroblastoma have elevated level of L-dopa. Occasionally, increased level of dopa is also seen in the patients with pheochromocytoma. What is pheochromocytoma? These are the benign tumors of chromaffin tissues in the adrenal medulla. And this dopamine then in the next step in the dopaminergic neurons by the enzyme hydroxylase, it is converted into norepinephrine, or in the adrenal medulla, this norepinephrine after transfer of methyl group is converted into epinephrine. So this is about the synthesis of the dopamine. The dopamine is formed in the dopaminergic neurons from the tyrosine, which undergoes first in hydroxylation followed by decarboxylation of dopa to form dopamine. And one important point to be noted is this, that these catecholamines cannot cross blood-brain barrier. Hence, in the brain, they must be synthesized locally. In Parkinson's disease, there is local deficiency of dopamine. Then, after dopamine is formed, then this dopamine, it is then transported. It is transported. Where it is transported? It is transported in the vesicular granule for storage. Therefore, dopamine, which is formed in the cytoplasm of the dopaminergic neurons, it is then transported in the vesicular granules for storage. And these vesicular granules, they are very smaller in size. They are approximately 50 to 100 nanometers in diameter. So now, how this dopamine formed in the cytoplasm is transported to the vesicular granules for storage. So how dopamine formed in the cytoplasm of the dopaminergic neurons is transported. Where it is transported? In the vesicular granules for storage. So you see here, this dopamine, it is transported in the vesicular granules of the neurons by the transporter, and this transporter is called VMAT2, vesicular monoamine transporter 2. So dopamine formed in the cytoplasm of the dopaminergic neurons, it is then transported in the vesicular granules for storage by the transporter, which is called vesicular monoamine transporter 2. Actually, there are two transporters. One is vesicular monoamine transporter 1 and vesicular monoamine transporter 2. Vesicular monoamine transporter 1 is expressed in the endocrine glands only. Vesicular monoamine transporter, it is expressed only in the endocrine cells, while vesicular monoamine transporter 2 is expressed in the neurons. So vesicular monoamine transporter 2 is responsible for packaging of DNA in the synaptic vesicles in preparation for synaptic release. Now this vesicular how this vesicular monoamine transporter transport this dopamine. So for this transportation, this vesicular monoamine transporter, it requires driving force for transport of dopamine from cytosol into vesicle. And so this driving force is provided by the ATP. So actually this vesicular monoamine transporter 2, it is ATP dependent. That is, ATP provides driving force for the transport of dopamine in the vesicle. And this vesicular monoamine transporter 2, which utilizes energy in the form of ATP for the transportation of dopamine. This vesicular monoamine transporter 2, it also transports hydrogen ions. Or this vesicular monoamine transporter 2, it also pumps protons in the vesicle to maintain electrochemical gradient between the cytoplasm and the vesicle. When these protons are transferred in the vesicular granule, this produces acidic environment in the vesicle. And this acid environment of the vesicle or this electrochemical gradient, it is very important for the storage of dopamine in the vesicle. Disruption of this gradient results in the rapid and massive loss of dopamine from the storage vesicle in the neural cytoplasm. And this disruption of this electrochemical gradient, it results this disruption of gradient, it occurs in the situation when there is ATP depletion, because energy in the form of ATP is required for transportation. So disruption of the gradient occurs when there is ATP depletion or when there is energy depletion, such as in the condition of ischemia, or when there is deficiency of blood supply in the blood vessels, or in the condition of anoxia, where there is total lack of oxygen, or in the cyanide poisoning. These vesicular stores of catecholamines are this vesicular store of dopamine. It is not present in the static state, but these vesicular stores are dynamic, are in dynamic equilibrium with surrounding cytoplasm. That is, there is passive outward leakage of dopamine in the cytoplasm, and it is counterbalanced by inward active transport under control of this transporter, vesicular monoamine transporter 2. So how this is released, we have explained you this, that this storage, it occurs in the synaptic vesicle. Entry of the dopamine is driven by pH gradient, which is established by the protein present in the vesicular membrane, that is vesicular monoamine transporter 2, that pumps proton in the vesicle at the expense of ATP. This whole process is of transportation is explained to you in detail already. And then there is dopamine, how dopamine is released. So dopamine is released from the vesicle by exocytosis. Release of dopamine from vesicle in the synaptic cleft is by process of exocytosis. And this process of exocytosis involves fusion of vesicle with presynaptic membrane. In the resting state, a single quanta of transmitter is released. What is single quanta? It is the amount equal to 10,000 molecules of the transmitter. Our single quanta means this corresponds to contents of one vesicle. So in resting state, this single quanta is released spontaneously, and this release results in the increasing the permeability of sodium in the cell, and there is depolarization. So in this way, the release of dopamine from the vesicular granule is by the process of the exocytosis. Then the dopamine receptors, after release of dopamine, there is binding of dopamine with receptors in the post-synaptic. So after release, there is binding of dopamine to its post-synaptic receptors. Now, there are at least 5 types of the dopamine receptors. These 5 types of dopamine receptors, they are classified in 2 groups. One is D1 like family and second group is D2 like family. So these 5 dopamine receptors, D1, D2, D3, D4, D5, they are divided in 2 groups. One is D1 like family. This D1 like family, it includes 2 receptors, dopamine receptor 1 and dopamine receptor 5. D1 and D5, they are referred as D1 like family, and these receptors are excitatory in nature, affecting adenylate cyclase positively. And second group is D2 like family, which includes dopamine transporter 2, 3, and 4. D2, D3, and D4. They are collectively called as D2 like family, and these receptors are inhibitory in nature, affecting adenylate cyclase negatively. So this is classification of dopamine. D1 like family, we told you, includes D1 and D5. They are excitatory in nature. And D2 like family includes dopamine receptor D2, D3, and D4. They are inhibitory nature. D1 and D5 excitatory receptors, they stimulate enzyme adenylate cyclase and increase the cyclic AMP. While D2, D3, D4, they are inhibitory nature and they inhibit enzyme adenylate cyclase. So this is, in this table, there is location and the function of dopamine receptors. Dopamine receptors, D1, they are present in the quadrate nucleus, putamen, olfactory tubule, hypothalamus, thalamus, in frontal cortex, and they increase adenylate cyclase. D5 are also present in the hippocampus, thalamus, and they also increase adenylate cyclase. D2 receptors, they are present in quadrate nucleus, putamen, and they decrease enzyme adenylate cyclase. D3 are present in the olfactory tubercle, they are present in the nucleus accumbens, and they also decrease enzyme adenylate cyclase. Similarly, D4, which are present in the frontal cortex, they also inhibit enzyme adenylate cyclase. And as far as the function is concerned, the D1 and D5 receptors, they function in the motor control, while D2 receptors, they are involved in the regulation of prolactin, the hormone of anterior pituitary, and D3 and D4 receptors are involved in memory, motivation, and emotional response. So this is about the classification and the functions of the dopamine receptors. Then coming to reuptake of the dopamine. The dopamine, which is released in the synaptic cleft, from the synaptic cleft, this dopamine is taken back to the cytosol of the surrounding cells. And this is again achieved by high affinity transporters, which are present in the presynaptic membrane. And they also use these transporters, which are involved in the reuptake of the dopamine. They use energy in the form of ATP for uptake. So if you see here in this slide, reuptake of dopamine, how dopamine is taken back. This is done by the transporter, and this transporter is called DOT. DAT, DOT. What is DOT? DOT is dopamine active transporter. This dopamine active transporter, it is integral membrane protein. that removes or this dopamine active transporter pumps dopamine from the synaptic cleft and deposits back into cytosol of the surrounding cell. This dopamine active transporter provides principal mechanism for rapid termination of signals in the neuronal transmission. This dopamine active transporter is the principal or important regulator of cytoplasmic dopamine under normal physiological conditions. Dopamine active transporter transports extracellular dopamine back into nerve terminals, and this recycled dopamine can again be incorporated in synaptic vesicles and can be reused as neurotransmitter. Then coming to the drugs. Some drugs, they interfere with the neurotransmission. How drugs interfere in the neurotransmission? These drugs can affect synapse at different sites. They affect synapse at variety of sites in variety of ways. So these drugs affect synapses in different ways, such as either these drugs, they increase the number of impulses, or these drugs, they cause release of neurotransmitter from the vesicle with or without impulse, or these drugs act by blocking the reuptake, or they block the receptors, or some drugs, they cause increased production or decreased production of neurotransmitters, or drugs act by preventing vesicle from releasing the neurotransmitter. So, in this context, cocaine is a substance that increases the amount of dopamine in the brain's reward circuit, and all drugs of abuse target the brain's pleasure center. All drugs of abuse increase dopamine, and this cocaine is a substance that increases the amount of dopamine in brain's reward circuit. Cocaine prevents reuptake of dopamine, and this cocaine satisfies the natural reward system in a big way. But repeated drug use also raises the threshold for this kind of pleasure. What does this mean? This means you need to take more to get the same high. Meanwhile, drugs make your body less able to produce dopamine naturally, and this leads to emotional lows when you are sober. Then other drugs, this methamphetamines, they also increase the amount of dopamine in the synapse in two ways. This drug enters the dopamine vesicle in the axon terminal, causes release of neurotransmitter. In this way, it increases the amount of dopamine in the synapse. And this drug also blocks the dopamine transporter for pumping dopamine back into the transmitting neurons. So by these two mechanisms, they increase the amount of dopamine in the synapse. Then coming to degradation of the dopamine. The enzymatic degradation of dopamine into its inactive metabolite. What is inactive metabolite of the dopamine? This is homovanillic acid. So, degradation of dopamine into homovanillic acid, it is carried out by two enzymes. That is catechol-O-methyltransferase and MAO, this is monoamine oxidase. This degradative action can be performed by monoamine oxidase isomers also. Isoforms of the monoamine oxidase are monoamine oxidase A and monoamine oxidase B. It should be noted that catechol-O-methyltransferase, this enzyme is predominantly expressed in the glial cells. In the neurons, this enzyme, COMT, that is catechol-O-methyltransferase, either it is missing in the neurons or it is present in a very low levels. Monoamine oxidase B, MAOB, this is mainly found in the astrocytes, whereas monoamine oxidase A predominates in the catecholaminergic neurons. This monoamine oxidase first converts the dopamine into 3,4-dihydroxyphenyl acetaldehyde, which is then degraded into 3,4-dihydroxyphenyl acetic acid by the action of enzyme aldehyde dehydrogenase. So dopamine is converted into dihydroxyphenyl acetic acid by the action of two enzymes, monoamine oxidase and aldehyde dehydrogenase. Then this 3,4-dihydroxyphenyl acetic acid, then by the action of enzyme catechol-O-methyltransferase, it is converted into homovalenic acid, which is then excreted. And in the other process, this dopamine, it is first converted into 3-methoxytyramine by the action of enzyme catechol-O-methyltransferase, and then by the action of enzyme monoamine oxidase, and by the action of enzyme aldehyde dehydrogenase, it is converted into homovalenic acid. And this homovalenic acid, which is the degradative product of the dopamine, is excreted in the urine. This monoamine oxidase enzyme is found in the neuronal tissues, and it is also found in other tissues like gut and also in the liver. In neurons, this enzyme functions as safety valve to oxidatively deaminate the dopamine to its and inactivates the excess of the neurotransmitters that may leak out of synaptic vesicle when neuron is at rest. So monoamine oxidase, it is functioning as safety valve, that is the neurotransmitter which leak out in the synaptic vesicle when neuron is at rest, it is converted into inactive form by the process of oxidative deamination. And then there are some drugs which are called as the inhibitors of enzyme monoamine oxidase. The monoamine oxidase inhibitors permit neurotransmitter to escape degradation, and they accumulate in the presynaptic neuron, and they leak into synaptic space. And this neurotransmitter causes activation of serotonin and norepinephrine that may be responsible for antidepressant action of these drugs. Then coming to the functions of the dopamine. So dopamine is performing multiples of the important functions. Dopamine is involved in cognition. Dopamine, which is present in the frontal lobes of the brain, controls the flow of information from other areas of the brain. Disorders of dopamine in the frontal lobe leads to decline in neurocognitive function, especially affects memory, attention, and problem solving. Then dopamine is also involved in the voluntary movement. Voluntary movements. As you know, the part of the brain which is called as basal ganglia. Basal ganglia regulates movements. Basal ganglia, it depends on certain amount of dopamine to function at its peak efficiency. When there is deficiency of dopamine in the brain, the movements become delayed and movements becomes uncoordinated. On the flip side, if there is an excess of dopamine, then brain causes the body to make unnecessary movements such as repetitive tics. Dopamine has also important role in pleasure and reward-seeking behavior. Dopamine is a chemical that mediates pleasure in the brain. This dopamine is released during pleasurable situations and stimulates one to seek out pleasurable activity. This means the food, sex, and several drugs of abuse are also stimulants of dopamine release in the brain. Dopamine has also role in the memory and learning. Levels of dopamine in brain, especially in the prefrontal cortex, help in the improved working memory. However, this is a delicate balance. As level of dopamine increase or decrease to abnormal levels, memory suffers. Dopamine has also role in the attention. Dopamine helps to focus and attention. Dopamine may be responsible for determining what we say short-term memory based on immediate response to certain information. Reduced dopamine concentration in the prefrontal cortex are thought to contribute to attention deficit disorders. Then dopamine also functions as inhibiting the production of anti-pituitary hormone prolactin. Dopamine is main neuroendocrine inhibitor of secretion of prolactin from the anterior pituitary. Dopamine is produced in the arcuate nucleus of hypothalamus. Arcuate nucleus is that part of brain which is directly above the pituitary gland. Dopamine produced by the neurons in the arcuate nucleus of the hypothalamus are released in the hypothalamic hypophyseal blood vessels, which supplies dopamine to pituitary gland. This dopamine, which is produced by the neurons in the arcuate nucleus, act on the lectrotrophs. So dopamine produced in the arcuate nucleus acts on the lectrotroph cells that produce prolactin. These lectrotroph cells can produce prolactin in the absence of dopamine. Dopamine, therefore, it is also called as prolactin inhibiting factor, or it is also called as prolactin inhibiting hormone. And what is prolactin? Prolactin, it is a polypeptide hormone which is composed of 198 amino acid. This prolactin, it is responsible for milk production. During pregnancy, prolactin secretion is increased and stimulates the growth of breast, but high level of estrogen and progesterone secretion prevents the milk production. Therefore, prolactin is unique among the pituitary hormones that it is under predominant inhibitory control of hypothalamus. In hypothalamic destruction, prolactin level may be elevated due to impaired transport of dopamine in the pituitary. Therefore, hyperprolactinemia can produce galactoria, that is, excess production of milk, or causes inappropriate secretion of milk and hypogonadism. Apart from this, dopamine is performing also other functions. Dopamine is one of the neurotransmitter which is implicated in the control of nausea and vomiting via interaction in the chemoreceptors trigger zone. Dopamine, which is produced in the retina, transmits signals for vision and helps to increase sensitivity to color and contrast when exposed to bright light conditions. Dopamine, which is produced in the olfactory bulb, transmit signals for smell. Also, dopamine in low doses brings about vasodilation. In many forms of shock, when there is already intense peripheral vasoconstriction, dopamine in small doses helps by reducing vasoconstriction. But dopamine in higher doses activates alpha-1 adrenergic receptors to bring about peripheral vasoconstriction like norepinephrine. Dopamine is also synthesized in the pancreas, which may act to reduce production of insulin. In this way, dopamine is performing many important functions. And then what happens when there is deficiency of dopamine? So when there is deficiency or when there is low dopamine level, you might feel reduced ability to feel pleasure. Personality becomes flat, bored, apathic, and have low enthusiasm, depressed, feels difficulty in getting through the task when interested. Person feels difficulty in paying attention and concentration. There is slowing of thinking, or there is slowing to learn new ideas. The person is prone to addiction. There is low libido or impotence. Mentally or physically, person is fatigued and sleeps too much and troubled when getting out of the bed. So these are all the deficiency symptoms of the dopamine. Then we are coming to dopamine imbalance. So when there is deficiency of dopamine, functional depletion of dopamine in the substantia nigra causes Parkinson's disease. So deficiency of dopamine causes which disease? Parkinson's disease. And overactivity or increased concentration of dopamine has role in the development of schizophrenia. So this Parkinson's disease is very important from the examination point of view. Parkinson's disease, this condition is named by a physician who described shaking palsy in 1917. And what is Parkinson's disease? It is degenerative disease. Causes motor and non-motor dysfunction and this results in the marked decrease in the synthesis of dopamine due to degeneration of cells in the small nuclei of the brain, which is called substantia nigra, and in the quadrate nucleus and in the putamen. So this Parkinson's disease, what is Parkinson's disease? This is degenerative disease. This disease causes motor and non-motor dysfunctions and this results in the marked decrease in the synthesis of dopamine due to degeneration of cells in the dopaminergic neurons, that is in the substantia nigra, in the quadrate nucleus, and in the putamen. Cells in these areas produce dopamine as neurotransmitter. The amount of dopamine released is proportional to number of surviving cells. So dopamine, it is major transmitter of nerves that interconnects the nuclei in the basal ganglia. And basal ganglia, we told you, is the part of brain that regulates movements, and it depends upon certain amount of dopamine to function at its peak efficiency. Lowering of the dopamine raises the ratio of acetylcholine because level of acetylcholine is not affected. This imbalance contributes to various disorders of the movements which are seen in the Parkinson's disease. This Parkinson's disease, it is characterized by resting tremors, which usually disappear during voluntary movements. There is bradykinesia, there is slowness and poverty of movements. There is muscular rigidity and postural inability. There is impairment of postural imbalance, which leads to disturbance in the gait. But this Parkinson's disease, it rarely occurs before the age of 40 years. Now, why the dopaminergic neurons in the substantia nigra are damaged. So, why dopaminergic cells in the substantia nigra are damaged is, in first place, it is not clear. There may be some genetic susceptibility in some cases. In part, the cell damage may reflect process of aging. It is observed that about 13% of the cells of the substantia nigra are lost per decade from the age of 25 years and onward. It is estimated that the signs of the Parkinson's disease appear when level of dopamine in the nigrostriatal system is dropped by 80%. So, aging is one of the important cause of loss of dopaminergic neurons. 13% of the cells in the substantia nigra are lost per decade from the age of 25 years and onward. But the symptoms appear when approximately 80% of the neurons in the substantia nigra are dropped. Then viral infection can cause Parkinson's disease, but it is relatively minor cause of Parkinson's disease. Exposure to high level of manganese also causes Parkinson's disease. Some drugs also cause Parkinson's disease, like reserpine or neuroleptics or antipsychotic drugs. They are important in causing Parkinsonism. Reserpine depletes dopamine by inhibiting dopamine storage in the presynaptic cells, and many neuroleptic drugs, they block dopamine receptors. And a dramatic outbreak of Parkinson's disease occur in young drug addicts using MPTP. What is MPTP? This is methylphenyl tetrahydropyridine. This methylphenyl tetrahydropyridine, this compound is byproduct in the production of heroin. It causes acute Parkinson's disease in the intravenous drug abusers. Then excessive production of hydrogen peroxide by the action of enzyme monooxidase. Monooxidase in its action of the enzymatic action produces hydrogen peroxide, and this H2O2, this is major factor of neuronal degeneration in the patients with the Parkinson's disease. So then in the Parkinson, there is deficiency in the activity of complex 1 in the electron transport chain. Then protein misfolding is also protein misfolding and aggregation. This is also one of the important cause associated with the Parkinson's disease. So these are some of the causes where there is damage to neurons in the substantia nigra. The neurons in the substantia nigra which produce dopamine. These are the 6 cardinal features of Parkinson's disease. One is resting tremors, rigidity, flex posture, bradykinesia, hypokinesia, loss of posture reflexes, freezing phenomenon. The diagnosis is by 2 of the features, one at least being resting tremors or bradykinesia. And then coming to treatment of the Parkinson's disease. The most widely used treatment of Parkinson's disease is the administration of DOPA. What is DOPA? Dihydroxyphenylalanine. This DOPA is precursor of the dopamine. If you remember the synthesis of dopamine, we told you tyrosine undergoes hydroxylation to form DOPA. And DOPA then decarboxylation is converted into dopamine. So this DOPA is actually used in the treatment of Parkinson's disease. This DOPA is converted into dopamine in the brain and various parts of the body by enzyme decarboxylase. Why DOPA is used? Why not dopamine? DOPA is used that because DOPA can cross blood-brain barrier. L-DOPA is used in spite of dopamine because unlike dopamine, L-DOPA dihydroxyphenylalanine is capable of crossing blood-brain barrier. Therefore, dopamine itself is not used, but precursor of dopamine is DOPA is used. DOPA crosses the blood-brain barrier, then it undergoes decarboxylation in brain to produce dopamine, which is important for controlling the voluntary movement, or it is used for the treatment of the Parkinson's disease. Then coming to schizophrenia. Schizophrenia, it is mental disorder reflecting disturbance of thinking, feeling, and behavior. Cause is unknown. Various psychological, social, developmental, environmental, genetic, and biochemical factors have been evoked. Biochemical observations regarding role of dopamine in schizophrenia. Increased amount of dopamine is observed in brain tissues sample of the patients. There is increased homovalenic acid. Homovalenic acid is the degradation product of the dopamine. So there is increased amount of homovalenic acid observed in the CSF of the patient. Normally, homovalenic acid produced from the degradation of the dopamine is excreted in the urine, and level of dopamine D2 receptors appears to be increased in brain of the patient with schizophrenia. And this is in the schizophrenia, there is elevated level of dopamine in the brain of schizophrenic